We're going to talk about quantitative trait loci, and there are four big steps that are involved in the QTL analysis. So the first one is the identification of the parents. So it's really important that the parents that you use in your to generate the populations that they are contrasting in the phenotype that you are interested on, and because some of the variation that cause that difference in the phenotype, it's because genetic. So basically we're going to try to identify what is the genetic component that it's caused because the difference in the genome between both genotypes. And then we have to generate a population. So there are different kind of populations, and when we have the population, we have to genotype and phenotype that population. So to genotype the population, we can use a different kind of molecular markers, and then we have to identify the regions in the genome that have association with the phenotype of a we are interested on. So the first we are gonna discuss about the parents and the population. So in this graphic, I just want to show you what happened after several generations of selection for the same cycle. So as you can see here, after artificial selection in the lines, we're going to increase the mean of the population or decrease the mean of the population in the phenotype that we're working on. So this it's really important that when we're selecting the parents to use for QTL analysis, the difference between the phenotype of them, uh, it's really different, because as bigger is the difference between both genotypes that we're going to use as parents, we're potentially we're gonna discover the regions that are related with that phenotype. So here we have the parent 1 and the parent 2. So we have to cross those lines to generate the population. And remember here that the variation that we can see in the population, it's because the meiosis that occurs, the recombination that occurs in meiosis between the parents that we use to generate the population. So then we have to use molecular markers. So we can use a different kind of molecular markers. The most popular, at least in the last 10 years, has been AFLPs, SSRs, and SNPs. And the difference between them is that in AFLPs we didn't need to know the sequence of the in the genomes, so we just use restriction enzymes to cut the DNA, identify the variations in a year between the samples. Then we started to work more markers as SSRs, that basically we need to know the sequence because we need to design the primers and amplify in a PCR machine the fragments and basically run that in the population, this kind of molecular markers. And nowadays, as I mentioned previously, we're working with SNPs because they are really abundant in the genome. There are changes in just one nucleotide in the genome, and that is why they are so abundant. And basically, yeah, the molecular markers are just uh, difference in the genomes that have a location in the genome that we're working on. And with the molecular markers, we can do a like representation of the chromosomes, and those representations are called a linkage map. So here, for example, we have a chromosome 1, and basically we can use the information of the molecular markers that we have in the population, that are these black arrows that we have here on the right. And here, for example, I have 5 different uh, individuals with different recombinations. So basically what we can do with the molecular markers is identify the recombinations, and with those recombinations we can estimate how close or far are those markers between them. So if there is no recombination between markers as between marker 2 and 3 in this example, so that means that those molecular markers should be close or closer that, for example, here marker 1 and marker 4. And again, so we try to follow the recombinations in the population, so that is why we have to estimate the frequency of recombination, and that frequency of recombination is basically what is the frequency that occurs one recombination between two markers. So basically we have to take into account in which individuals of the population we have recombination, and we have to divide that with the population size, and with that we have the frequency of recombination. So in this example we have a progeny, a population size of 100, so the offspring is, the size of the offspring is 100, and in just one line we got a recombination, so that as you can imagine that frequency of recombination is 1%. And but if you are familiar with QTL analysis, we usually don't talk about frequency of recombination. Instead of that, we talk about centimorgans. And the centimorgans is the map unit, or basically the generic distance between two molecular markers. So to estimate that generic distance, we use the frequency of recombinations, that in this case is R, and we use different functions. So the most common are the Haldane and Kosambi. Haldane assumes that there is not a inference between one recombination and another one, so there is not effect. If there is one recombination, there is that recombination doesn't affect another one in the chromosome. And the Kosambi assumes that there is interference, so that one recombination in the chromosome can affect another recombination. So as you can see here, I don't want you to remind this formula, but all the elements in the formula are constant number. The only number that if you are interested to use those functions are the R. And as you can see here, if we replace the R with the R frequency of recombination that we got in our example, you can see that the centimorgan, or the centimorgan is equal to 1.01 centimorgans. That basically is the same that I mentioned here, that the recombination frequency is equal to 1%. And if we use Kosambi, again, all the elements are constant values, but the R. And if we replace the R here, you can see that the value with Kosambi of centimorgans is equal to 1 centimorgan. So that means that we have one individual that have recombination for in the 100 individuals in the population. And that is true, so Haldane and Kosambi are equivalent, but only until there are not more than, or they are, it's not bigger than 30. So after that, if you want to use the formulas and do yourself those or estimate the centimorgan with 30 or more, 0.30 or more, you can see that Kosambi, it's gonna give you a lower centimorgan distance or genetic distance. And that is why as Kosambi assumes that there is interference between recombinations, so it tried to correct in some way, or try to better estimate the genetic distance between markers. And that is why Kosambi it's one of the most common functions to estimate centimorgans in QTL analysis. And okay, and the last part of the first steps, the last step is the analysis of the QTLs. So in basically, like, this is a good graphic to see what happened if we have one QTL in the population. So here you can see that when we have when we have a QTL with strong evidence, it's when the means between the mean of the population between the individuals that have the are homozygous for little m and the mean of the individuals that are homozygous for big M, it's really different. So we can easily see that there is a difference if we have one genotype or the another one. And when that difference between means is not so evident, the evidence of that QTL, the presence of a QTL, is not that good, or it is not that easy to identify. So just to give you a better understanding of that, we can use this example. So just suppose that we're interested in seed size, and we have one parent that have a phenotype of 10 grams and another parent that have a phenotype of 20 grams. So we can generate an offprint, and in the F2 we're going to have that all the individuals that are homozygous for little m are equal to 10 grams, all the individuals that are homozygous for big M are equal to 20, and all heterozygous are 15. So as you can see here, the mid-parent value that we estimated in a previous video, it's the point that it's between the parents. So we have basically the average of the phenotype of the parents is the mid-parent value. So here we have that the mid-parent value is 15, and that value is equal to the heterozygous lines. So that suggests that this gene has an additive effect, and there is not evidence of dominance, because remember that the dominance or dominant deviation is which the difference between the midpoint of the homozygous lines and the heterozygous. So and with those values we can basically estimate which is the difference between the lines that are homozygous for both for big M or little m, if we know what is the additive effect. So what is the effect in the phenotype of, in this case, each big M, and if we know what is the recombination that we have between the markers. If we know which one is the recombination, yeah, it's the recombination in the population, and the additive effect, we can estimate the mean, the difference between the homozygous lines. So as you can see here, when the recombination is 0.5, that is the highest recombination that we can get. So this value here is going to be equal to 0, and the difference between, and if this part of the formula is 0, so the difference between the homozygous lines are going to be equal by 0, equal to 0 as well. And it's going to be equal to 0 because that means that if the recombination is 0.5, it's that the marker and the QTR are not a link. So that means that that marker doesn't have any association with the QTR. So that is why the expected difference between the homozygous lines is equal to 0. So because there are not association. So just to give you the same example but with some numbers, so here we have the additive effect. So each time that the genotype increase a big M in the genotype, the phenotype increase 5 grams. And yeah, we can use this formula. So if the recombination is equal to 0, so that means that if R is equal to 0, so that means that there is no recombination between the marker and the QTR. You can see that this 2 times 0 is going to be 0, so this part of the formula is going to be equal to 1, and in that case we can estimate that the difference between the homozygous for big M and homozygous for little m, it's going to be equal to 2 times the additive effect. So as the additive effect is 5, we can estimate that the difference between a homozygous for little m and homozygous for big M is going to be equal to 10. That is what we can see here in the graphic. Okay, so that is why it is important to estimate the recombination. And there are other approaches maybe that are easier to understand to estimate if one marker uh, has association with the phenotype. And here again we can do like the t-test that basically compare the mean of two genotypes, so in this case the both homozygous, or which is more common, it's in QTL analysis, it's use linear regression. So why linear regression is because in linear regression we can estimate the p-value to know if one molecular marker uh, has association with the phenotype, but also we can estimate the R square. And with the R square, we can, that is additional information that we can get with the linear regression. Basically, we can estimate how much of the variability of the phenotype is explained with the genotype. And just to give you an idea about the R square, if you are not familiar with this term, it's basically here if we have a high R square, that means that the that the independent variable can explain most of the variability in the dependent variable. So here we have, for example, the genotype, and this genotype can explain almost 0.1 of the variability of the phenotype that is in the y-axis. And when the R square is lower, so you can see that the independent variable cannot explain very good the dependent variable. So that it's an additional information that is useful for the interpretation of the QTL analysis. And there are basically two like big categories for QTL detection. The first one is the single factor analysis that basically use one marker at a time, as the example that we discussed previously, and basically identify the difference among the means of the genotypes. But there are two main limitations with single factor analysis, and the first one is that the location of the QTLs cannot be determined. So here, for example, you can see that we have 4 molecular markers and we have one QTL here. So maybe we can detect that the QTL, the marker number 2, it's associated with this QTL, but we have no idea. That is the only thing that we can identify with a single factor analysis. We cannot identify if that QTL is upstream or downstream. So that is a limitation. And another limitation is that in this example we have two QTLs, and now we have a association with two markers that have association with these two QTLs, but we don't know if those markers are associated with independent QTLs. That in this case they're associated with two different QTLs, or another possible result of this single factor analysis is that both markers are pretty close and they are linked to the same QTL, but we cannot basically uh, conclude if we have one QTL or two QTL. So as you can see here, that is a big limitation, like the location of the QTLs and the number of locations that we can detect with single factor analysis. And that is why, like the most common example, or most of the studies work with interval mapping. And there are several interval mapping approaches, and I just want to mention two of them. Maybe they are the most common, that are the simple interval mapping, that basically use adjacent markers to identify the QTL. So as you can see here, the QTL is here around 20 centimorgans, and we have the marker Q and the marker R, and as they are adjacent each other, so we can define that the QTL is here, because the strongest association of the molecular markers and the phenotypes, it's in this region. And as you can see here, the marker P and S are not so very associated with the phenotype, so that is why we can define that the QTL it's in this region. But there are some additional issues with interval mapping, and that is why it's the composite interval mapping is pretty useful to try to identify like with a better resolution the location of the QTL. So composite interval mapping use also the interval mapping, but in this case this model also use markers that are in the background of the genetic map, as covariables in the model, and basically that allows this model to identify as like more, the identification of the QTL with composite interval mapping is more reliable because it use more information in the genetic map. And as review in QTL analysis, so I want to mention that it's really important use parents that are contrasting in the phenotype that you are interested on, so that it's probably the most important step. Select two parents that are really different in the trait that you are interested on. You have to generate the population, and then you have to collect the phenotype in that population and run some molecular markers in that population. And then you have to create the genetic map. Remember that the genetic map, it's basically a way to um, group the molecular markers by chromosomes. And, and here it's important to point out that as bigger is your population, the resolution of your genetic map is going to be higher. And when I say resolution, is the number of recombinations that you are going to have in your genetic map. Because yeah, with bigger population, you are going to test, or you are going to follow a bigger number of recombinations in your population. So that is good, because that is what we want to basically identify, like a lot of recombinations to try to identify molecular markers that are linked to the phenotype that we are interested on. And if you are looking, and if you identified one molecular marker, like in this example, that is linked to the phenotype of interest, so you can use that molecular markers as in early generations to do a marker assisted selection. And if you can use a molecular markers to select the germplasm, you can reduce the cost, and you can also increase the efficiency of your breeding program. So this is not easy. We are going to discuss about the implementation of QTR results later into, yeah, in the next video. And But this is the idea. So in traits as a resistant to diseases, for example, in common being we have a couple of markers because there are like some major QTRs that control most of the variation in the phenotype, and we are gonna discuss that later. And this is the end of this video.